This is part 3 of the gram-negative cocci lecture. And in this lecture, we are going to focus on laboratory diagnosis of Neisseria. So, meningococcus, or MC, or Neisseria meningitidis suspected specimens must be processed in a biological safety level 2 cabinet, or a laminar flow hood. And in this biosafety level 2 cabinet, the air is being moved in the cabinet so that it's going to maintain any aerosols inside the cabinet so that they don't come out and accidentally expose you. The Neisseria gonorrhea and Neisseria meningitidis require CO2. So you're going to incubate the specimens in a carbon dioxide incubator as opposed to an ambient air incubator or an anaerobic incubator or jar. So gonococcus, or Neisseria gonorrhea, you have to use chocolate agar, or you have to use selective media specifically for Neisseria gonorrhea. For meningococcus, or Neisseria meningitidis, you can use 5% sheep blood agar, you can use chocolate agar, or you can use a selective media. So meningococcus is a little easier to grow. Now, there are selective media that are very commonly used to isolate Neisseria gonorrhea, and these selective media include modified Thayer-Martin agar, Martin-Lewis agar, and New York City agar. So in the first lecture, I had lecture notes on Blackboard. In those lecture notes, I tried to include a chart of all the different media that we're going to discuss throughout the rest of the semester. And you'll see that that chart tells you what the media is, what organism it's used to isolate, the ingredients inside that media that either make it selective or differential. So you should, when we talk about a specific media, you should go back to those lecture notes from week 1 and find that media and highlight that media and memorize a bit about it, because the exams in micro will include questions. You are expected to know some things about the selective media. The board exams also will ask you questions about media, because we as medical laboratory scientists are expected to know, if I think I have this group of organisms, I need to know what media they can and cannot grow on and why. So you definitely need to know about modified Thayer-Martin agar. It's an incredibly important media, and I would say most clinical laboratories use Thayer-Martin to streak out all genital samples in order to rule out Neisseria gonorrhea. For these media, what makes them selective is they have antimicrobials in the media. So commonly they have vancomycin in there, and that is going to not allow your gram-positive organisms to grow. They commonly have colistin in them, which is going to not allow some of the other gram-negative organisms aside from your Neisseria and to grow. And then it also contains an antimicrobial called trimethoprim, which is going to inhibit the growth of Proteus. Proteus is a gram-negative rod that commonly will overgrow plates, and you won't be able to find the organism you're looking for if you have Proteus on there. So these are 3 antimicrobials that are commonly found in selective media for Neisseria. These media will also have antifungal agents in them to reduce any fungi from growing, such as nystatin, anisomycin, and amphotericin B. Here is a picture of Neisseria gonorrhea growing on a modified Thayer-Martin agar plate. Now the thing about modified Thayer-Martin is it looks exactly like chocolate agar. So when you're working in the laboratory, all of these media are commercially purchased, and usually they have a little stamp on the bottom of them of an expiration date, a lot number, and what the media is. So you always want to be really careful to, especially with a genital specimen, you're commonly going to use, in some labs you might use a blood agar, a chocolate agar, and a Thayer-Martin agar. In some labs you may just use blood and Thayer-Martin, so it's all going to depend. But if you're using chocolate and Thayer-Martin, or even just the Thayer-Martin, you need to make sure it's a Thayer-Martin and not a chocolate agar plate. There is also a media called Gembec that's similar. It looks again like chocolate agar. It's a square plate that has a little cover on it, and commonly you would inoculate in a Z pattern. So the Neisseria, gonorrhea, and meningitis are somewhat fastidious or fussy. They like CO2, so they're going to grow better in a CO2 incubator. Gonococcus or Neisseria gonorrhea is going to die relatively quickly if it's outside of the body. So the specimen has to be delivered to the laboratory in specific media so that the organism doesn't dry out and die. The organisms prefer 35 to 37 degree body temperature range. They produce small glistening colonies within 1 to 2 days of incubation. Now the non-pathogenic Neisseria are non-fastidious. They're not fussy at all. They'll grow on standard commercial lab media. They are aerobic. They'll grow in an ambient air incubator. They'll also grow in CO2. They'll grow at room temperature. So the pathogens, the pathogenic Neisseria, are much more fussy than the non-pathogenic Neisseria. All of these organisms are gram-negative diplococci. They are flattened on one side. They look like a coffee bean or a kidney bean in their shape. And the non-pathogenic organisms tend to be larger than the pathogenic organisms, although they'd have to be side by side under a microscope for you to really see that. Commonly, they are found intracellularly in a clinical specimen, so in a spinal fluid or in a male urethral discharge, you will see a lot of white blood cells, and you'll see the gram-negative diplococci both intracellularly and extracellularly. These are non-motile organisms. They do not form spores. So to diagnose gonorrhea, usually from a male specimen, it is a direct diagnosis of the urethral discharge. You do a gram stain of it, you see a ton of white blood cells, you see the gram-negative diplococci, there you go. The patient has gonorrhea. You don't really need to move any further than that. Now the key with gonorrhea is you can diagnose directly from a male urethral specimen. You cannot do that in a female specimen. So if you get a female cervical swab in, you cannot just do a direct gram stain. You have to actually plate out that swab onto your non-selective and selective media, incubate it, wait at least 24 hours, and then work that organism up. And the reason is Females have a lot of different normal flora in the genital urinary tract. Some of the normal flora in the female genital urinary tract look very similarly to Neisseria gonorrhea. You can see gram-negative cocci and even diplococci in a female genital specimen. And so you can't automatically assume that it's Neisseria gonorrhea. So for males, direct smear, gram stain, you can diagnose Neisseria gonorrhea. Females, you cannot. You have to grow the organism up and do biochemical tests on it. For meningitis, usually, like we said, the specimens are going to be cerebral spinal fluid and blood. You may also get a skin lesion. Commonly, Neisseria meningitis causing meningitis is going to be diagnosed based on the symptoms. So an individual had a severe headache and a stiff neck and now has a rash, a petechial rash all over their body and is lying in a coma. That is very clearly Neisseria meningitis. They will likely be treated for Neisseria meningitis before any specimens even get to the laboratory. So here is Neisseria gonorrhea, the white blood cells that you will see in the intracellular and extracellular gram-negative kidney bean shaped diplococci. So we are going to move on to the fourth part of the gram-negative cocci lecture.